What's up, YouTube? Welcome back to Logical, Plausible, Probable. In this video, you're going to get to see the annihilation of a supposed biochemistry expert who is doing research in the lab that is showing how close we're getting to proof of origin of life from the atheist perspective. But it turns out they don't actually have the confidence, or at least when they get finally called on the carpet, can't actually back up their claims, even though for 10 minutes they say that I'm making things up and don't know what I'm talking about. So make sure you stay tuned to the end where he admits that he has been making things up. But before we dive into it, make sure you like this video and subscribe to the channel. Make sure you click the little bell icon, otherwise YouTube won't send you notifications when new videos like this one go live. And I really appreciate you sharing this with your friends as we attempt to spread the word about how the atheist and evolutionary perspective of how we exist is neither logical, plausible, or probable.
Someone also mentioned evolution, and as it's my area of expertise, I thought that might be a useful thing to unpack again. But if it's not strictly relevant to the conversation, we can do that another time.
That's going to be John's call.
I'm okay with it. The evolutionary component has significant relevance in the atheist worldview, whether or not it's an accepted premise or not. Because if it is completely invalid, especially in the context of going to abiogenesis, then the whole premise.
Are we talking
the whole premise of? No, I'm saying that both Darwinism, going all the way back to abiogenesis, is a significant foundational point for the atheist worldview. If either one is false, especially abiogenesis.
If I can interject,
the worldview is a fantasy. Go ahead.
Do you know the Catholic Church position on evolution?
I do, but that has zero relevance to the point that I'm making from a scientific perspective. I mean, I have debates on this exact topic, and I could care less about what the Catholic Church position.
Yeah, that's totally valid.
Yeah, considering the Catholic Church stance on pedophilia, we're not going to trust their stance on evolution.
Yeah. So. Yeah, that's fair. Abiogenesis, though, is a different thing, is the term that you have as well.
What? I missed the last portion.
Prebiotic chemistry is a very specialist field, and like emerging into biochemistry is a really interesting field.
Oh, I mean, it's a very interesting one, and it's growing. It's becoming a more and more. The goalpost is getting further and further away the more we learn about biology, and this is a pretty freely admitted premise, even by the Lee Cronins of the world, when he's actually put on the spot about it. That we have no clue, and he's considered one of the leading origin of life researchers.
Well, that's not close to prevailing opinion. Put it that way.
Oh, really?
Okay. What aspect do you think is the major issue? Which aspect?
All of them. So you've got you've got genetic code, you've got the protein problem, you have the replication problem, you have all of you have all of the pieces of biology that. I mean, you have the homochirality problem, you have the homeolinkage problem, you have there's an ongoing list of recognized issues from a prebiotic chemistry perspective that are freely admitted and force a utter reliance on deep time for this to even have a remote premise of plausibility. So, I mean, to say that this is not something that's in major conundrum, I'd like to find out where you're getting that from, because there's ongoing stuff from this year talking about how they're nowhere close.
Well, it's interesting. You link the problems that you have, as expressed in a list, to deep time. Is that the key issue?
No, I'm saying that you're you are the one that has to rely on deep time. I'm saying that deep time, without deep time, the premise of abiogenesis is not even a remotely feasible component. That's freely admitted, even though the improbabilities of it completely destroy the entire premise of deep time in the first place.
Yeah.
Because so I'm not really following what your point is.
Yeah, I completely accept the requirement for extended periods of prebiotic chemistry. And we have those. So it would take a first-year lecture course to go into all of them, but this is what I do for a living, and it's essentially what I've been doing for all of my career. The apparent problems that you have seem linked to perhaps a young Earth position that is itself very difficult to establish as fact.
I haven't said anything about young Earth.
Correct.
I'm talking about the origin of life. And I would like you, since you supposedly do this professionally, I would like you to point me to a paper or your own professional stance that says we actually have a remote clue other than pure hypothetical. Oh, wait, I guess if all of these things happened perfectly, then please expound, because I read up on this stuff on a regular basis. And everything I'm seeing, and I was reading papers on this last night, actually, and they from this year, and it's still being freely admitted that it's not. So you're the expert. Go ahead. Multiple, but go ahead. You're the expert. Tell us how we're so close.
Precise question.
You just said a few minutes ago that you professionally are doing this, and that we are very close to understanding how this is done. And you said you do this professionally. So please expound. How so?
Well, what part of abiogenesis is the problem in terms of the chemistry that you see being the first point? That it nucleotide analysis? Is it catalytic based on matrix? Is it self-replication? Is it membrane formation? Is it ribozyme activity? I mean, any of these?
So I'm pretty sure that all of those represent problems, as you well know, and the organization of all of those into a working system is another layer of that issue. Correct?
Well, the things you've described as problems are all demonstrated to be.
So you're telling me that the helicase. So you're telling me that the helicase. You're telling me that the helicase can form itself. So you're saying the helicase can form itself.
No, I'm telling you that polynucleotides.
Are you saying that a ribosome? Can a working function of a ribosome form itself?
That with this at all. So I'm trying to understand where the
is a ribosome an ultimate component of transcription and translation and protein synthesis.
What you've said there is meaningless.
It's meaningless. So you're saying that a working ribosome is not
because those things can happen in a ribosomal.
By themselves, you're saying that the full.
What I'm saying is, are you saying that full
protein synthesis can occur without a working ribosome and all the other proteins and enzymes that are required for the transcription and translation process?
Well, it seems that you're moving questions each time.
I'm not. I'm asking the ultimate question of abiogenesis, which is how does a living system come into existence without all the parts? Ultimately, especially in terms of the replication process, which there's many, and it's been recognized in quite a few papers that the entire premise of the full replication system being able to come into existence all by itself is not really a plausible deal. I mean, are you telling me that the linkage problem just for nucleotides is not a valid one? Because I'm pretty sure like the last paper I saw on it was like around 200. It starts to collapse.
So are we talking about protein synthesis or nucleotide polymerization? Because those aren't the same thing. No.
Well, I'm saying that all of these are relevant. So I'm saying okay. you're saying that
they all are.
Okay, so is the linkage problem valid? Yes or no?
Which linkage? Protein or DNA or
nucleotide.
RNA?
Nucleotide
linkage. A phosphodiester bond is what is the question?
The question is in the lab, do we have a bonding problem that usually, in the range of around 200, where a artificially created strand starts to collapse?
I see.
Is that yes or no?
We're now talking about helicase activity in the presence of high temperature, right?
No, I'm saying without the helicase. I'm talking about just in context of synthetic DNA.
So at this point, you've taken me down a rabbit hole of about 19 questions.
This one's a pretty direct. If you're actually an expert, this is a very direct one, because this is a fundamental component of biogenesis.
Perhaps rephrase your question and summarize it in a sentence.
Okay. In order for DNA to form, you have to have the base pairs, correct? And they have to bond to the phosphate backbone, correct?
Well, the phosphate backbone is one thing, and
there's more about
bonding to each other.
Right, with the hydrogen bond. Yes, with the hydrogen bond. What I'm saying is, when we've in the lab, when we try to create a synthetic DNA strand and bond and
by
link
do you mean
the nucleotides?
Or just...
I'm saying the experts in the chemistry lab, when they try to do it synthetically, not modifying existing DNA, they try to do it artificially. Around 150 to 200 nucleotides in length, it starts to collapse, and they have not been able to make it past that point in the lab.
Are you saying that we can't synthesize DNA beyond 200 nucleotides in the lab?
We can synthesize DNA that's when we're modifying existing DNA. When we're trying to build it completely from scratch, we have the homeolinkage problem.
I'm really sorry, but you're way behind. I've done this myself many times, and I'm sorry, you're just factually incorrect on that. But I mean, is there another thing you want to jump into? Maybe you seemed concerned about protein folding. And
wait a minute. Hold on. Wait a minute. No. You just said he was factually incorrect about something. Then correct him. You don't get to just ad hominem his argument and then say let's move on and think you're getting ahead over. No. And address his argument.
Okay. How would you like me to do that?
You said what he just said was inaccurate and wrong. I expect you to be able to elaborate, unless you're just trying to ad hominem the argument to get away from it without looking like you can't answer it. So I would appreciate you answering his argument instead of just attacking it.
So if I gave you a publication in which I've synthesized DNA multiple kilobases in length, would that suffice?
Sure.
100% from scratch.
From
scratch? Let's see that from the paper.
100% from scratch.
As in from atomic level or nucleotide?
You had free floating nucleotides, and you synthesized kilobase DNA strands.
Okay. Without an existing template?
Without an existing template, and without any enzymes or any of the transcription components.
So we've extended that from a PCR environment to an abiotic.
Have you done it without any of those components?
Environment. Well, not without components required to do the reaction. So I withdraw my comment.
So thank you for trying to BS your way through this and try to act like I didn't know what the hell I was talking about and claim that you have done this and solved this problem when you're literally full of shit.
You're correct. Bro. If you don't have the ingredients to make the cake, you'll never.
So what is abiogenesis, buddy? Do you have? This is the whole point. If you don't have those factors, then you are working from scratch, correct?
Well, I'm working from.
You're trying to work like it would be from prebiotic soup, right?
Sure.
Okay, so ultimately, what we have gone around and danced around for the last 15 minutes is that. No, we have no. You have not done what you claimed you have done in terms of a direct comparison to the prebiotic soup because you're not being able to do it without the factors that I delineated as being requirements in order for life to exist, which you then dismissed. So now we've eventually come back around to the reality that A, I was not full of shit. B, you were, and you were trying to completely issue a facade to present that I didn't know what I was talking about. Because you're Mr. Expert. But since you are the expert, supposedly, you knew full well that you were full of crap because I was directly asking you these questions.
Again, if you don't have the ingredients to make a cake, you'll never make a cake. And yeah, I was incorrect in what I said, and I hope you accept my apology for that.
Okay, so now we're going back to ribosomes, right? So we're now discovering that there's like what appears to be nervous system-esque circuitry in them, and they're being relevant to
which
protein synthesis.
Which kind of ribosome and which subunits are we talking about, or just ribosomes generally?
Well, in this context, ribosomes in general. I was reading a paper on this last night about how they're looking at the whole argument of ribosomes being purely mechanistic systems into having significantly more comprehensive information processing components in the circuitry. And to me, when you start to think about that of, you're now having to, in the abiogenesis argument, you're now having to conclude that it's somehow an extremely complicated 3D printing factory on the molecular level able to form itself.
Well, are poly peptides 3D by definition, or do they fold as a function of their environment?
What is the end result of the linear connection of amino acids? If it's done correctly, it folds into a 3D structure, right?
In certain environments, yeah.
Yeah, and then there are other instances, there are chaperone proteins that come along and do additional modifications and et cetera, right? But the end result is a 3D structure, correct? Okay.
Yeah.
So
absolutely.
How is that any different than a 3D printer? So the end result of 3D is taking a linear. It's taking a linear information data set, just like you do in 3D printing, and then turning it into a 3D output. I mean, that's exactly what a 3D printer is.
Yeah. Yeah, protein biochemistry is really cool that way.
Yeah, it's amazing. And the improbabilities of it happening by chance get pretty nuts, don't they?
In the absence of selective pressure, sure. But if something works and can reproduce itself, it will tend to proliferate.
Okay. Yeah. So if it reproduces itself and it has all the different components that are required for the reproduction, then maybe. But then you then have to get into the whole aspect of, if you don't have the mRNA, how does the ribosome work?
Again,
not in your hypothetical, not in your hypothetical world. In the real world, in the real world, does a ribosome does protein synthesis operate without an mRNA?
Well, obviously not. Can polynucleotides reproduce themselves through autocatalysis? The answer is some can do that.
Right. Well, heck, there's some pseudogenes in RNA that go and seem to execute themselves into proteins. In an abiotic. But those are completely but those are completely formed structures that already exist, right? Because if you don't have all the codon data set, then you're not going to result in a protein.
We're talking about nucleic acids, not proteins, in an abiotic environment, which can catalyze their own reproduction.
Right. But how does
a lot like viruses?
How do the codons assign themselves?
Okay, again, we're getting into an entire course.
You just went back to nucleotides. How did?
Yeah, I'm just talking about process in a biotic environment where reproduction with selective pressure can happen. It's really cool.
For DNA or for RNA?
For RNA at that point. Right.
So somehow you're saying that reverse transcription happened for the RNA to turn into DNA. Is that what you're saying?
No. Just chemistry.
Okay, right. So how did the RNA become the DNA?
Didn't necessarily have to. The point that I'm making is...
But we're now operating from DNA. So apparently from your worldview, it did happen. So how did it happen?
Let's see. How much time do you have?
That's another thing we don't have any real clue over, do we? It's all pure hypothetical.
Again,
because in biological systems, the way that RNA gets turned back into DNA is through reverse transcription, doesn't it? With proteins that are required to already exist, right?
No. DNA is turned into RNA typically in that way, but okay.
No, I'm talking about things like retrotransposons, where it's moved as mRNA and then translated back into DNA. That's what I'm talking about. So that's the only, and as far as I'm aware, that's pretty much the only time that RNA becomes DNA.
Did we switch? Did we switch then from abiogenesis to virology and retroviruses?
No,
that's an area too. I don't know.
So basically what we've gotten from all this is, number one, you were trying to act like I was full of shit when you knew full well that what the argument I was making was correct. You're now trying to act like I'm jumping around when I'm making the point that your arguments require these factors to come into existence at some point. And yet
you seem to be presupposing my arguments, really. What are my arguments?
Well, I mean, when I called you out directly, you've pretty much agreed with the points that I've been making.
Okay. All right. So tell me my arguments, please. Go for it.
Well, let's go back to questions. So how, in terms of the codon amino acid assignment, how did that happen?
So you're not going to tell me my arguments, are you? You're going to assert that you know them.
I just asked you a question. Did I make an assertion? I asked you a question. I literally just asked you a question. How did the codon amino acid assignment happen?
Okay, before we get into that, can I ask a question to understand the environment better? It's meant with good faith. What is third base wobble? Is that a concept you
I'm very familiar with third base wobble, and I'm also very aware of the fact that third base wobble has a major variable in the context of transcription pausing, or sorry, translational pausing. And I know that you're probably going to try and go down this whole, it was only it wasn't the triplet, it was the dual aspect, et cetera, et cetera. The question I would have for you in response to that is, if every single codon in the ribosome had a third position wobble, would a protein synthesize? Or is it is that basic information theory where there's a premise to account for data or noise in copying?
Again, are we talking about copying fidelity, or would a protein synthesize, or third base wobble? Could you maybe define what that is succinctly?
Why are you asking about third base wobble? Because I just responded to why your question really isn't that relevant to the points that I'm making in terms of the codon assignment. So go back to my original question of, how did codon assignment being equal to an amino acid occur?
Degeneracy within the codon assignment, right? As well.
Sorry?
Okay.
Are you just trying to dodge the question? You're supposedly an expert. So how did codons and the syntax of the code of the genetic code, how did it become equal to amino acids?
What is third base wobble? Could you explain it in a few sentences?
Answer my question.
No, not until you demonstrate this.
Base wobble, as I've already talked about, in the ribosome, there may be an error match in the tRNA, and it still enables the process to continue. There's more to it than that. That's one of the components of it.
Yeah, there's quite a lot more to it than that. So would third base wobble introduce the same amino acid, a different amino acid, or a similar amino acid? And I'm asking you for a shibboleth here.
It does I think the first one and the third one, you said, in some instances.
Okay.
But now, going back to my question, how did codon assignment happen to even be resulting in third base wobble that we're talking about now? How did the codon assignment ever occur?
How did it ever occur? Amino acids, but as a function of their chemical nature in the environment of the intracellular space, adhere specifically to individual tRNAs.
No. I'm talking in the DNA. How did the codon become equal in the DNA, being transcribed into mRNA, then being matched to a tRNA? I understand that whole process. I'm saying, how did it happen in DNA through the abiogenesis process?
It wasn't necessary in the immediate abiogenesis process. We're talking about dramatic times away from in the initial.
How did it happen? Period.
Biochemistry.
No. Okay. The codon never actually interacts with the amino acid. This is a well-known fact. Interacts with the tRNA and the anticodon. It never interacts with the amino acid.
Yeah, that's right. Yeah.
So how did something that does not chemically interact with the end result and end up being read by the ribosome to equal that? And then when there's errors in some instances like third base wobble, or if it's the wrong amino acid connected, even though it has the correct anticodon, it gets ejected. So how do you explain that? Because that sounds to me like arbitrary value assignment. How do you explain that?
The ones that could do it reproduced, thrived, survived, and flourished.
There is no life at this point. In order for life to exist, this has to already be accounted for. There is no life survived and flourished.
Okay, fair enough.
So let me ask you direct questions, because if you're not saying that would have to exist, then how did all of the transcription, translation, and replication processes that you're relying on, how did they happen without those variables?
Could I ask, at what point did you define what you mean by that? Because it's quite important to the discussion, I think.
How do you ever answer questions of, like, how do some of the most vital portions of what's recognized for life to exist come into existence without just trying to dodge it?
Okay, are prions alive? Are viruses alive?
Answer my question on life. But viruses, it's a who knows question. I would say no, because they lack so many major variables of what we recognize as life. But in the context of because are viruses actually reproducing themselves? Or are they leveraging these components that I'm talking about being required in order for life to exist?
It really depends on the virus.
So all you are is a dodger. How did the transcription process come into existence?
Very slowly.
Very slowly. So you're saying that a hyper complex level of molecular machinery came into existence very slowly.
Incrementally, yes. At no point did.
So are you
staggering
suggesting
complexity pop out?
So you're suggesting that the 1,000 amino acids that are required for a helicase, you're suggesting that's just somehow slowly formed when it's I mean, what was it being used for? If there was no DNA, if there was no protein,
it was very possible. It was very possibly a less good helicase. And
okay. So and are you saying the ATP is required for it to rotate? Are you saying that all that stuff just happened too?
I'm actually quite interested now in your alternative hypothesis to biodiversity.
No. You came in here as Mr. Expert talking mad shit to me, bro. You claim to be an expert, and you claim to have all these answers to this stuff. So you're now the expert, and I'm questioning you as the expert. I want to know. You said you do this professionally, so I want to know. And you also, at the beginning of this whole interaction, you said that we were very, very close, and my position, which was that no, we're not, was false. Mm-hmm. So you need to start answering questions about how this happens, not just over time. Aren't you? Are you a biochemist? So how are we creating? Can you recreate a helicase in the lab from scratch? You know the exact sequence. Can you do it from scratch by yourself? No enzymes, no transcription factors whatsoever. Can you recreate a helicase?
Well, I can't recreate it if I don't have
John,
any of
I'm sorry, guys. Just quick interjection. Can you repost?
Yeah.
We have requests, so can you repost the link in the chat? Sorry, guys.
Yeah.
And I'm pretty much done, guys. I've enjoyed the conversation.